In the very central, deepest part of yourself, I knew something was wrong.
It was unfathomable that this would be my fate.
It was a very tough, dark time in terms of trying to survive.
It's a shock. It's something that you just don't know how to deal with.
My dad finally came in. He sat me down and he said, you know, you have cancer.
Sometimes in science, we're not exactly wise. We allowed our definition of cancer to not evolve over 160 years.
We didn't really have any fundamental understanding about what cancer was.
It was a bit like trying to send a man to the moon without really understanding the laws of gravity.
Almost everything has changed about what we know about cancer.
We are fighting against 200 different diseases.
It's a very complex problem that we have trivialized and miniaturized to make digestible to the general public.
We are curing some people who don't need to be cured.
To feel that the medicine is just right there and you didn't have a chance to fight it.
How much we spend on weapons, on military, on fighter jets, and when you compare it to investment in cancer research, there's really an order of magnitude of difference.
We need a public movement.
This game of yards, let's move the yards. Let's play the violin in public. Everybody should know how we're doing.
There have been more advances in the last 20 years than in the previous 2,000 years. We know more about the disease now than we've ever known. So after all this time, after all this progress, the question remains, will we ever see a day without cancer?
angioClast is a drug delivery company with the aim of treating solid cancers. So what is unique about us is that instead of targeting cancer cells, which are variable and evolving, we're targeting the blood vessels supplying the cancers instead, which mutate less and are easily accessible. By using a library of markers that are exclusive to us, we can, for the first time, distinguish between cancer-associated vessels and those vessels supplying healthy organs. Now, we can then make antibodies against these markers and use them to deliver any compound straight to the vicinity of the cancer. This has huge potential in dramatically improving current treatment options and diagnostic strategies. One of the applications of this technology is that we can combine these antibodies with specific compounds to selectively destroy tumor vasculature. So we can cut the lifeline of the cancer and starve the tumor to remission.